So I'm going to talk about genomic diversity in modern humans, but I'm also going to have a look towards the past. And I was told I first of all had to put this disclosure, there's no conflict of interest here. So one thing I want to start with is that when we look at genetic diversity in modern humans, it's truly immense. And let me just put it in perspective. In 2014, we knew of 73 million single nucleotide polymorphisms called SNPs. And so those 73 million known SNPs could be put together in 10 to the 35 million combinations of genotypes. And that number is so big, to kind of put it in perspective, the cosmologists tell us there's 10 to the 80th electrons in the universe. Okay? So what this means is there's also other forms of genetic variation, like copy number variation. So the actual amount of potential genotypic variation in humans is literally astronomical. And there's only less than 10 to the 10th humans, and that means that every individual has a genomic combination that is truly unique in the entire history of the universe. It will never, ever be replicated. And of course, there's more mutations occurring every generation. Now, this genetic diversity has many medical applications. Genetic risk factors for systemic diseases, an area I've worked on quite a bit. Genetic resistance to susceptibility to infectious diseases. Genetic responses to environments, and that would include drugs and other kinds of medical treatments. Not everybody reacts the same way to the same treatments. We also know that the gene expression is very dynamic in how it responds to environment, and interestingly, much of this dynamic gene expression itself is under genetic control and is part of the genetic diversity that humans have. And finally, I'll be talking about evolutionary adaptation to environments, and in fact, this has very direct disease implications. Now, to make the use of this huge amount of genetic diversity, we need to, first of all, know how it is structured within and between human populations. And to do that, we need to understand the evolutionary origins of this diversity and understand how it's spread through space and time to create the current configuration of genetic structure in the human population. And there's basically two ways of doing that. Ancient DNA, which I'm not going to talk about because we already heard an excellent lecture about that in this hall just an hour ago. But I'm going to talk about the analysis of current DNA polymorphisms. And you might wonder how by analyzing the current DNA can we understand past evolutionary processes. Well, you all know about DNA replication, so you know how one molecule of DNA can produce two daughter lineages. Well, evolutionary biologists are often backward thinking. Okay, we like to look at the present and then look backwards, not forwards. And so if you look at DNA replication backwards, what you can see is that two different molecules of DNA today could have been one molecule in the past. And this backwards feature of DNA replication is called coalescence. In fact, if you look at any homologous piece of DNA in the human genome, and you look far enough back, there's one coalescent event after another until all the pieces of DNA that are present in modern-day humans coalesce to one ancestral DNA molecule. And that's true for every homologous region of the genome. And it turns out, therefore, because most of the time DNA replication is faithful, DNA itself, the current DNA, is in one sense ancient DNA. It's a mixture of ancient DNA and more recent mutations. And particularly in areas of the genome that undergo little to no recombination, we can reconstruct that evolutionary history with a great deal of accuracy. And we now know, because recombination is concentrated in hotspots in the human genome, we can easily find many, many regions of the nuclear genome in which there's little to no recombination, as well as mitochondrial DNA and Y DNA, which also don't have recombination. So we can use this to reconstruct the past evolutionary history of humans without the necessity of going to ancient DNA and fossils. Now, to do that, you need a lot of statistics. And don't worry, although my background is in statistics and I'm in statistical genomics, I'm not going to show any equations. Okay, I know that's puts everybody to sleep right away. But I did develop a technique called Multilocus nested clade analysis that extracts statistically significant associations of this evolutionary history of genes in these non-recombining regions and shows how they're associated in time and space and can interpret the patterns that emerge using this coalescent theory, this extensive theory in population genetics that deals with this backwards DNA replication. I want to emphasize that all retained inferences are based on the rejection of null hypothesis and have explicit quantified statistical support. And that's what differs this technique from many other ways. I think one of the big problems I've noticed since I work both in human genetics and non-human genetics is that quite often when we apply science to our own species, some of the criteria we use kind of go off to the side. Our own preconceptions of what the answers ought to be dominate. And the only way I know to protect ourselves against our own innate biases is to use objective statistical criteria. So the criteria I'm going to talk about today are the same criteria I use when I analyze elephants or lizards or whatever. We all share the same basic evolutionary processes. Another big advantage of this technique is you don't need a prior model of human evolution. For example, the last quarter century we've had what I would regard a needless debate between what is called out-of-Africa replacement and multi-regional. These were prior models of how humans evolved, and the debate was very ferocious and often nasty, and it was really based on nothing. It was really based on preconceptions. And so here you don't need a prior model. The model emerges automatically from the retained statistically significant inferences. And so here's the model that emerged. I first published this in a Nature article in 2002. Since then, I've added more data, obviously, refined the analysis, but the results have actually changed very little. And one of the things that you can see is there are 3 major out-of-Africa expansion events in recent human evolution over the last 2 million years. And the dating of these events is strictly 100% from the genetic analysis. It's not from anything else, yet it corresponds extremely well to other data. So, for example, at 1.9 million years, we have an expansion out of Africa into Eurasia. And this corresponds in the fossil record for the first expansion of humans from the genus Homo out of Africa into Eurasia, which was dated, now the oldest fossil is 1.85 million years. So it really corresponds beautifully to that. The dates of these 3 expansions also correspond to the 3 wettest periods in the Sahara region in the last 2 million years. And also areas where we could see from soil carbonate analysis, there was a big expansion of the savanna. And remember, humans were a savanna species. We evolved in the savanna in Africa. And the fossil record shows also many other savanna organisms expanded out of Africa at these same time periods. So this is very consistent with the fossil record, both the human and non-human fossil record as well. And then I could not reject the null hypothesis of isolation between the Eurasian populations and Africa, so you see them as separate lineages. But then the next event is a mid-Pleistocene expansion at about 700,000 years ago, which I call the Acheulean expansion. And like I say, this is compatible with the paleoclimatic data, also the archaeological data. The Acheulean culture first arose in Africa and then spread into Eurasia at about this time. And so then after that, you see that it shows a trellis-like structure because the analysis indicated there were genetic connections between Africans and Eurasian populations established. For some strange reason, many of the portrayals of human evolution show humans can only move in one direction. They can only move out of Africa. Well, if they can move out of Africa, why couldn't they move back in and back and forth? And in fact, more recent data has shown that overlaid on these major periods of wetness, there are minor fluctuations, and during the pluvial periods, archaeological data shows humans did inhabit regions that are now deserts. So there were many corridors opened up on a scale of tens of thousands of years that would allow genetic interconnections between Africa and Eurasia. So Africans and Eurasians are not genetically isolated, at least since the mid-Pleistocene. Then we have the final out-of-Africa expansion event, the one that everybody talks about, and that occurred at 130,000 years, and that's compatible also with the archaeological and fossil evidence that shows modern humans were in northern Africa, the Arabian Peninsula, and the Levant by 125,000 years ago, and recent fossil finds in China that shows anatomically modern humans were in eastern China by 110,000 years ago. So those earlier dates that you often see of 60,000 years just are not compatible with either the paleoclimatic data, the archaeological data, or the fossil data, or the genetic data as analyzed by Multilocus Clade analysis. After that, there's a lot of gene flow, and then more recently, there was expansions in the last 50,000 years into northern Eurasia, the Pacific, and finally the Americas. And one of the things I want to emphasize is just the admixture. The expansion of the Acheulean, you can reject the null hypothesis of no admixture of that expanding African populations with the earlier Eurasian populations, just barely. It's just barely significant. But notice the most recent out-of-Africa expansion, it's rejected at a p-level of 10 to the minus 17th. And this was from 2002. So we've known since 2002 that there was admixture between these expanding humans out of Africa with Eurasian populations. And this, of course, has been confirmed by the ancient DNA studies that we heard about earlier. But it wasn't a surprise. We've known that there was admixture, mostly out of Africa. The admixture rates were low, but we've known that for quite some time. Now, what are the implications of this model of human evolution? First of all, humans have evolved as a single evolutionary lineage for at least the past 700,000 years because of gene flow and admixture. It also means that genetic diversity in humans is primarily in the form of individual variation. I'm different from you, and that's where most of the genetic variation lies. And there's actually very little differentiation between human populations. Now, most people are very surprised when they hear this because we look at ourselves and we think we're very different. Different human populations from different parts of the world appear to us to be extremely different. Well, one of the nice things about genetics is we can quantify that in an objective way and actually see it. And since I work both on human and non-human species, I have a very different perspective. In fact, humans are about the most homogeneous species on the face of this planet. And I'll just give one example. Here's a study that was done on chimpanzees, the common chimp. And there's 3 subspecies that are recognized. In the red, the blue, and then the orange and green is now one subspecies. The purple is a different species of chimpanzee. So those are the 3 regions or subspecies of chimpanzees. And AMOVA is just a way of quantifying how much of the genetic variation in the species is seen as between population differences. And you can see in chimpanzees, it's about 30% of their genetic variation is between these 3 major populations. Now, we also can look at humans on a global level, and so I've circled the 5 populations that were in this study. And there we can ask how much of the human genetic diversity is found as variation between these populations. So we're on a much bigger geographical scale than the chimpanzees, and you can see it's 4%. Think of that. Okay, in fact, even if you add the Neanderthals and the Denisovan individual, they are also not as different as the modern subspecies of chimpanzees. So humans at a global level, 95% of our genetic variation is just due to the fact that we're individuals. And very little is due to the fact we have, quote, different populations. Now, there's many implications of this. You may have noticed that in that model, most of the gene flow in humans, the genetic interchange, fit what we call an isolation by distance model. Because genes can replicate, you don't have to have, say, an African going all the way to China to have gene flow between Africa and China. Rather, they just interbreed with nearby populations, and each population is a stepping stone. And the gene, of course, by replicating, can take many generations to go across that boundary. So, in fact, China and Africa can be united genetically by these stepping stone or isolation by distance models. And in fact, we find that much human gene flow, even today, fits an isolation by distance model. And this also means that geographical distance is the major predictor of genetic differentiation between human populations. So here's just one study that was published a few years ago in PNAS, and it plots a genetic distance between pairs of human populations on one axis with the geographical distance using waypoints on the other to constrain most movements across land as opposed to going across oceans. And you can see that, in fact, most of the genetic differences between human populations, that small 4% on the average, is actually explained by geography. Geography is the single most important predictor of how different two human populations are, even though those differences are very minor. Now, even with isolation by distance limiting gene flow, I don't want to say that genetic differences between human populations have no significance, because even that is enough limitation to allow local adaptation to occur in human populations. So even though we show very little overall genetic differentiation, there are some genetic differences that are important adaptively and also clinically. And I'll just give a couple of examples. One is sickle cell anemia. At least in the United States, this is often treated as an African American disease. But some people of, say, Greek ancestry are often misdiagnosed, or their diagnosis is late because, of course, sickle cell is an African disease, not a European disease. But if you actually look at the distribution of the sickle cell allele, you find it's really an adaptation to malaria. In areas where malaria has been particularly virulent and causing high mortality, we have high frequencies of sickle cell. So sickle cells in Africa, it's more widespread in Africa because malaria is, but it's also, as you can see, found in Europe and the Mediterranean region. It's found in the Arabian Peninsula, and it's found in Asia in the Indian subcontinent. So it's not an African disease, and by making it that, we've actually, I think, done a lot of clinical harm to many people. It's a disease associated with malaria, and many local adaptations in humans follow this pattern. They don't follow what we preconceive as racial lines or continental boundaries. They really reflect local environmental conditions. Now, another local environmental condition that I'm going to talk about now relates to skin color. And this is some research we've recently done on the vitamin D receptor gene and the skin color genes. And it involves several people. We have three joint co-authors, senior co-authors, Dov Tiosano, who is here, Charlie Klimmer, and our president of this meeting, Laura Adi. And I put Charlie in bold because she's the one who came up with this idea of a new computer program called Blockbuster, which I and Wingxiao Zhang helped modify a little bit, but she's the real genius behind it, that is so efficient it can scan all pairwise combinations, literally of millions of SNPs. We've already, and look for associations between specific alleles all across the genome. We've already applied this to some disease research, but I'm going to talk about it now with respect to skin color. So if we look at skin color, we see a pattern probably most of you know. Dark skin is mostly in the tropics, light skin is in the higher latitudes. And when we look at skin color, we see a cline. We see a very strong clinal distribution of skin color. And I want to emphasize that skin color is associated with latitude, not racial category or other things. So I'm going to look at two individuals who are from this one population, which you can see has one of the highest genetic differences at the molecular level. So here's one woman, and here's another woman. One of these women is from sub-Saharan Africa. The other one is from the island of Fiji. Now, a European woman, being geographically intermediate between these two, would be actually genetically closer to both of these women than either of these two women are to each other at the molecular genetic level. So this is an easy one. I've chosen two women from two of the most genetically distant populations in the world. Which is the African, which is the Fijian? Nobody wants to guess. Okay, so the woman on this side, which would be your left, is the Fijian. The woman on the far side is the African. And I'm sure you all got it right since this is such an easy one to guess. So I want to emphasize, when populations are interconnected by gene flow, adaptive traits do not always reflect patterns of movements of peoples or individuals in their evolutionary history. Rather, they reflect the local adaptive conditions. Now, one of the interesting things that we revealed with Blockbuster is that although any individual skin color gene or the vitamin D receptor often does not show a clinal distribution, we found significant associations at the multilocus level, even between genes that are on different chromosomes, alleles that came together in a complex. And when we plot that complex, which is shown in the solid dots, it fits a nice linear cline with latitude. And in fact, this explains over 80% of the variation. So we do see clinal variation as well, and that's another attribute of human variation. Now, because of isolation by distance, admixture, adaptive clines, and local adaptation like we saw with sickle cell, we find that human individuals do not cleanly belong to a single population category. Again and again, people say, well, which population do you belong to? Given our evolutionary history, I think that's a meaningless question. Rather, we belong to a hierarchy of relatedness among all humans, and not just a single population affinity. So very recently, we've tried to develop a new approach to encompass that, and we're using network theory to study population structure. And actually, this is no longer in prep, it's just been submitted. Gilly Greenbaum is a doctoral student at Ben Gurion University in Israel. His undergraduate degree is in mathematics, and he's really just been brilliant. So he's been the primary developer of this program. And I and Shirley Bar-David, who's at Ben Gurion University, are his co-doctoral advisors. And the approach is to start with individuals as the units of analysis, because that's where most of the genetic variation is in humans, between individuals, not between populations. All individuals are then interconnected in a network by an allele-sharing genetic distance measure, and we've come up with a new one which we think has better properties than some of the other ones out there. And we then look exhaustively at all distance thresholds. That is, you come up with a threshold and you ignore all connections that are below that threshold. And you keep doing this, and at each stage, we calculate what is called the modularity. That is, we look for modules within the network where most of the interconnections are within that module and few between, and they're statistically significant. And that way we can identify all the emergent groups at that level. And we go through the entire range of possible thresholds. And we applied this approach to 11 HapMap populations. We chose 1,000 SNPs per chromosome so that we didn't have to worry about correlations within the chromosome. So for a total of 22,000 SNPs. And actually, we ran this on a desktop computer. And so this is a very computationally efficient program. Other programs that allocate genetic variation, like we started structure in August on this, and it's still churning out, and this is on a supercomputer. So here are the results. When you have a threshold of less than about 18%, there's one global human population. We're all interconnected very strongly. Like I say, compared to chimpanzees, we're very homogeneous. When you get above 18%, we now split between, and that's part C down there, We now have the sub-Saharan Africans and the rest of the world, and that reflects our evolutionary history of these out-of-Africa expansion events. And then as you increase the threshold to almost 19%, you split the Western Eurasian one you split the out-of-Africa populations into the East Asian and the Western Eurasian populations. And then when you increase the threshold even more, the Western Eurasians split into Europe, India, and Mexicans. And the Mexicans here have a very strong Western European component, which is why they're clustered. And then finally, if you increase the threshold more, you separate the Chinese from the Japanese. And above that, all population structure disappears. Now you're really finding relations between individual relatives, even though these populations were chosen not to have known relatives. So that really reflects the hierarchy of human populations. All these populations are real. There's not a single population to which we belong. You can also, with this, assign to each individual in the sample a strength of association. So when they're assigned to a module, how strong is it? And one of the things you see is that the Western Eurasian, the Asians, and the Africans have very different distributions of their strength of association. And this is another big difference. Many of the programs try to find populations instead of each population is the same. Here we can see they're not the same. And we can get a lot of insight by dividing them into the HapMap populations. So for example, with the Western Eurasians, we see the two European populations are homogeneous and have a very strong unimodal strength of association. The Mexicans, on the other hand, as you see, have a bimodal distribution, reflecting the fact that they're an admixed population between American Indians and Western Europeans. And the Indians show a broader distribution, showing they've had repeated gene flow from many other populations, including Western Europeans. If you look at the African, it's bimodal, and you also see a little red dot. Those are the African Americans. Once again, the admixture sticks out very strongly. The Maasai population is that outlier in the rest of Africa, and it shows a pattern, and I'm not going to show all of this, that indicates a fair amount of gene flow over long periods of time with other populations. So we can get a lot of insight, and in fact, we can identify specific individuals who are admixed to certain degrees. So now that we're in the genomic era, I think we really need computationally efficient tools to make use of the immense amount of human genetic diversity that we have in our search for genetic associations and responses to personalized medicine. And the focus really needs to be on individuals, because that's where most of the genetic diversity exists. So a lot of us paid lip service to the idea of personalized medicine and the role genetics plays in it. But what we really need to do is get away from the idea of races and populations and so on, and focus on the individuals and their individual genetic diversity. That's where the future of personalized medicine lies. Thank you.